Hello, my name is Curtis Eckerman, and in this video I'm going to show you how to take the multiple sequence alignment that we did in the last video and turn that into a tree, into a cladogram or a phylogenetic analysis. And last time we looked at the sequence alignment between 4 dog species, or 4 of the canids, including a coyote, a dog, a wolf, a jackal, and that of a mountain lion. And again, we included the mountain lion as an outgroup. And we did the sequence alignment, and it gave us this distance matrix. To construct a tree, there are different algorithms, different mathematical calculations that can be used to determine which sequences belong together, or which ones are more closely related versus those that are more distantly related. One of the most common algorithms is something called neighbor joining, and we see that in the form of this BioNJ and these others. Actually, all of these have some variation of neighbor joining with some tweaking of the particular algorithm, or some kind of a variant, if you will, on that. We're going to use something called PhyML. The ML stands for maximum likelihood, and it's using the calculation to determine the most likely of the various trees, and it's using a philosophy that we've talked about, in the sense that the shortest the tree with the shortest distances on it, that is those that have the fewest changes, should be the most likely. Of course, there's a lot more math involved in that process. You can also use bootstrapping. Bootstrapping refers to doing this many times from different starting points, and the idea being that if you do this many times, you can actually get some statistical analysis, but that takes a while, and it's beyond our purposes here in the lab. So we're going to use PhyML, and I'll tell you right off the bat that if I used any of these, in this case I would get very similar trees, if not identical trees, because this is a pretty simple analysis given the fact that the sequences are so similar and that I'm very confident in the alignments themselves. So I'm going to choose PhyML. I could actually select this at the bottom of this screen as well. I could do it down here as well instead. Doesn't really matter. Whatever way I do it, I'm going to click on execute. Now this takes a few moments to do. This one is not too long. Other ones can be quite a bit longer depending upon how many species are involved, how long the sequence is, because it's having to do several calculations as it groups sequences of similar length together, and continues to do that calculation. So this one's over, and as I scroll down, I see my tree. All right, now what I want to do is I want to change a few options here, because this tree looks a little confusing at first. If I were interested in branch lengths, I could be using branch lengths based on the number of differences and how much change has occurred. I can use that for a variety of different reasons, but again, it's beyond our lab here. So I'm going to click on the options of without branch length and without the branch length labels. And so I get a nice tree here showing us the relationships of these 5 organisms using something called mid-root outgroups. Now, normally we would use the puma or the mountain lion here as an outgroup, and we are, but we didn't designate it as an outgroup. Instead, the calculations are looking at this mountain lion as a really distantly related organism, and it's rooting it at the midpoint between this really distantly related organism and the others that are closely related. And so I get a nice tree here showing the relationships of the 4 canids that I have here, the dog, the wolf, the coyote, and the jackal, compared to a not so distantly related cousin here, the mountain lion, who's also in the order Carnivora, but is in a different family, the Felids, the Felidae versus the Canidae. And so that's how we use an outgroup to kind of give direction, if you will, of changes. And so in this analysis, we would see the jackal and the coyote as being more ancestral than the wolf and the dog in this case. Now that may or may not be the case. You would have to probably add more species to this to get a more finer detail of what's going on in the canids, but there's a few things of note here, in particular that I get a pretty decent relationship. I clearly see that Canis lupus and Canis lupus familiaris, which based on their name we would suspect they're closely related, do cluster out as being the most closely related. We also see that the dogs as a whole, the canids as a whole, cluster together away from the cat. That's information that we would expect just simply looking at the physical features of these organisms. So the fact that we get the same relationship based on molecular data tells us that indeed ancestral information, genealogy information for species, if you will, is captured in the molecular data. That should be very encouraging to us all, given that we can reconstruct phylogenetic histories using these molecular markers. And so at this point, I'm feeling pretty good about the tree. It shows me some relationships that I definitely expected to see. Maybe I don't know exactly what the relationship should be between Canis aureus and Canis latrans and the others, but I do see two indicators that tell me that there's good information here. One, that Canis lupus clusters together, and two, that the canids cluster together outside of the felids. All right, so now I need to download this particular tree. I can click on PNG. This is a photo format. I can click here and download it to my desktop, or I can use a snipping tool. All PCs have a snipping tool. Macs have something similar to this. I can click on, actually I'm going to see my tree. I can click on the snipping tool, click on new, and I can highlight my tree. And I've just now copied my tree. I'm going to make this smaller so that we can see this a little bit. Or I can see the whole thing. I've captured this. I can paste it. I can copy and paste it. I can save it. And I now have a picture of my tree. I'm not going to save it here. I will tell you, though, the easiest thing is just to download a PNG of your tree. SVG is a vector diagram. This is a more detailed image and allows you to zoom in really closely on it. It's certainly going to be a bigger file, and it's not necessary for our purposes. A JSON tree can be used in other types of programs for analysis. Again, for our purposes, the PNG is going to be just fine for that. So you would download your tree, and this is what you would upload into your worksheet once you've done the worksheet exercises. So that would be a similar way of doing that. Okay, we have taken now, and I'm going to zoom out just a little bit just to see everything. We have taken some data from GenBank. We have used ClustalW to align the sequences that we downloaded, and we have now taken that information, that alignment, and turned that into a cladogram, a phylogenetic analysis of the canids using a mountain lion as our outgroup. And I think the results are really well demonstrated here. I can tell you that if depending upon what groups you're looking at, sometimes this can be very complicated, but for our purposes, this demonstration purposes, turned out very well, and we would be very happy with our results. Okay, so that goes from downloading data to the final analysis, and this is how you would do that using some very useful online tools.